Good morning to my supervisor, Professor Dr. Emil, and examiner. I am Yew Zhi Yan and would like to present my final year project 1 entitled Gene Sequence Analysis of Tumor Susceptibility Gene 101 TSG101 in Nasopharyngeal Cancer Cell Line. As for the introduction, TSG101 Tumor Susceptibility Gene 101 is a human gene that maps on chromosome 11. According to Lee and Cohen, the TSG101 was found from mice and has 3 T3 fibroblasts through an approach known as the homozygous knockout approach. Besides, most research also indicate that TSG101 may have a variety of function in carcinogenesis as well as the development of tumor in diverse cell types. In addition, nasopharyngeal cancer is a cancer that originates at the upper parts of the throat and behind the nostril. According to Chen et al., NPC is epithelial cancer that develops mostly from nasopharyngeal mucosa as well as was frequently observed in pharyngeal lesions. As per the International Agency for Research on Cancer, roughly 129,000 new instances of nasopharyngeal carcinoma were detected in 2018, representing only 0.7% of all carcinoma detected in that year. Problem statement. According to Chua et al., TSG101 deletion 154 mRNA generate an increase of TSG101 protein in human nasopharyngeal carcinoma biopsies, which was replicated by overexpressions of TSG101 deletion 154 in the NPC cell line, TW01. However, the research on mutations such as insertion, etc. of the NPC cell line is still limited. Thus, further study on the gene sequence analysis of TSG101 between NPC cell line and normal cell line are required to understand the relationship between the TSG101 gene in NPC cell line and normal human cell line, as well as to observe the other types of mutation or genetic lesion in the TSG101 gene in the context of NPC. The objective of my FYP project is to analyze the gene sequence of TSG101 between normal cell line MP69 and NPC cell line TW01 and CCC61. Hypothesis. TSG101 was also found a mutation in human papillary thyroid carcinomas. Thus, the hypothesis of my FYP project is the gene sequence of TSG101 is expected to be different between the normal cell line and NPC cell line. For the methodology, it is divided into 7 parts. For the cell culture, 2 NPC cell line and 1 normal nasopharyngeal epithelial cell line in the culture flask will be provided for RNA extraction. Next, for total RNA extraction, the triazole methods will be utilized to extract total RNA from the human nasopharyngeal epithelial cell line as well as the NPC cell line. While for the quantification and integrity check, the UV spectrophotometer at the absorbance of 260 mN and the ratios of the absorbance at 260 and 280 mN will be used to determine the concentration and purity of the RNA extracted. 2 UL of RNA will be pipetted out and dilute in 48 UL of autoclaved distilled water and then analyzed by UV spectrophotometer. The A260, A218 ratios of 1.8 to 2.1 was projected to obtain a sufficiently highly purified RNA. Next, forwards and reverse primer for TSG101 will be designed using the NCBI primer design tool, BLAST. Then, the design primer will be forwarded to empirical scientific Senjian Berhad for primer synthesis. Then, the reverse transcriptase of RNA will be conducted according to the protocol provided by Promega. Moroni murine leukemia virus transcriptase will be utilized to synthesize cDNA from the TW01, C6661, and MP69. PCR of TSG101 from the 3 cell line will be performed and multiplied to obtain a precise result. For aggressor gel electrophoresis, each of the PCR products will be loaded into each round. The amplified TSG101 gene will be excised from the aggressor gel using a sterilized blast. The excited amplified TSG101 will be sent to DNA sequencing with the gel image and 5 UL of both forwards and reverse primer to empirical scientific Senjian Berhad. Next, the sequence of TSG101 received from the empirical scientific Senjian Berhad will be analyzed using the blast to observe any presence of a mutation or nucleotide variation between the gene sequence of the 3 cell line. In addition, a multiple alignment program, MAFFT, will be used to compare the similarity as well as differences between these 3 sets of gene sequence. At the end of this study, I expect the gene sequence of TSG101 in the human normal cell line to be different from the gene sequence of TSG101 in the NPC cell line. TSG101 was often mutated in human breast tumor, as well as its aberrant splice variants were found in a variety of tumor types. Therefore, the gene sequence of TSG101 in the NPC cell line is predicted will have the mutation. This is the expected work schedule of my project. I expect to conduct cell culture on November and December 2021, total RNA extraction on December 2021, quantification and integrity check on December 2021 and January 2022. Primer design and RT-PCR on January and February 2022. Aggressor gel electrophoresis on February and March 2022. DNA sequencing on March and April 2022. While report writing and presentation will be expected to conduct on April and May 2022. These are the references. That is all from me. Thank you and have a nice day.